Arthropods that host parasitoids are not entirely passive victims, and parasitoids have evolved some very unique ways of manipulating host behavior to improve their own fitness. In this lecture, we will explore some of the ways hosts can defend themselves from parasitoids using both physiology and behavior, and some of the ways that parasitoids can manipulate host behavior. In addition, we will touch on the subject of non-consumptive effects, the various ways that fitness of prey species can be reduced by the presence of natural enemies, even if they are not killed and eaten. Parasitism entails intimate physiological interactions between host and parasitoid, the outcome of which is often uncertain. The focus here is on insect parasitoids, not fungi and not parasites that do not kill their host, as the ecological implications are very different. For example, parasites experience selection to attenuate their impact on host physiology because the continued survival of both host and parasite are linked together. This is only true to some extent for coinobiont parasitoids that must grow with their host for some period. The more specific the parasitoid is to a particular host, the stronger their co-evolutionary relationship is likely to be. But this does not preclude the existence of host defense through behavioral and physiological means. It is also important to remember that not all changes in host phenotype resulting from parasitism are necessarily a consequence of direct selection. However, incidental byproducts of parasitism that benefit parasitoid fitness can evolve into various forms of host manipulation. A number of examples in this lecture will come from aphid parasitoid systems, simply due to my greater familiarity with these systems, not because they are necessarily more representative of these phenomena. Host defense can take three forms. The distinction between chemical and physiological defenses being that chemical defenses take the form of secretions outside the body of the host and before parasitoid oviposition. Physiological defenses are those mounted by cellular processes within the body of the host subsequent to oviposition by the parasitoid. The most basic behavioral defense of an aphid is to drop from the plant, but this behavior is highly variable across aphid species. Some species feed with their stylets deeply embedded in plant tissue and are therefore unable to withdraw them quickly without sustaining harm. More generally, the fitness consequence of dropping from the plant entails costs that are very context dependent and vary with the probability the aphid can regain the plant again. There is also an intrinsic cost which is not insignificant for such short-lived organisms: the loss of feeding time and thus reproductive success while the aphid is off the plant. When the cost of this behavior is too high, aphids may engage in more aggressive defenses, such as kicking at the parasitoid or trying to smear it with cornical secretions. These secretions, which you see here in this exuded from this Aphis nerii, act like a fast-drawing crazy glue. They're extremely noxious to parasitoids and potentially fatal. These droplets also contain E-beta-farnesene, the alarm pheromone of aphids, which serves to alert conspecifics to the danger. However, their secretion is not without some physiological cost to the aphid, and there is also an indirect cost because the alarm pheromone also serves as a chiromone that attracts aphid predators. A single cornical droplet can seal the mouthparts of an early instar coccinellid larvae, preventing it from ever feeding again. More aggressive aphids, like the black bean aphid here, will actively try and smear the attacker. In this video, which in full length takes over 20 minutes, the female wasp gets her antenna stuck on the actual cornicle and is dragged around by the aphid until she collapses and dies from exhaustion. This has no long-term consequence for the aphid, which will shed the wasp during its next molt. Physiological defenses against parasitism are much more difficult to study, as you would expect, but they are no less important. Encapsulation is a process whereby special hemocyte cells are recruited to the parasitoid egg immediately after oviposition. These cells employ a process of melanization to form a hard capsule around the egg or early instar larva that prevents it from breathing or feeding. Because there is a quantitative element to the encapsulation, it tends to be more effective when employed by large hosts against smaller parasitoids. However, more virulent parasitoids can use venom-like proteins to shut down the host immune response, whereas more resistant hosts can prevent this. It really becomes a genetic arms race in evolutionary time. Here is an illustration of such an interaction, comparing the responses of two Drosophila species to two strains of the parasitoid Leptopolina boulardii. Resistance in both host species is conferred by one major locus with two alleles, whereas either line of the parasitoid can successfully parasitize Drosophila melanogaster when it is homozygous for susceptibility, on the left. Only the line co-adapted to this species can parasitize the host when it is homozygous resistant. In contrast, even the line co-adapted to Drosophila jacoba can only parasitize this host when it is homozygous susceptible, and the resistant allele is not required to defeat the non-co-evolved parasitoid. These pictures from the same publication show the various stages of encapsulation in Drosophila. This article is included in the module for reference only. You will not be tested on all the detailed biochemical pathways involved. Within 12 hours after oviposition, hemocytes can be seen depositing melanin around the egg. Once killed, the egg remains as an inert object within the body cavity that is visible in all subsequent life stages. As with many such antagonistic interactions between insects, outcomes can vary depending on the relative strength of the two individuals, their physiological condition, and other factors such as the speed of the melanization response. Here, the larval stage has been achieved prior to completion of the encapsulation process, and the young larva has been able to survive by breaking out of the capsule. The red arrow indicates the mouthparts of the larva. Among entomopathogens, host manipulation is often used to maximize dispersal of reproductive propagules. We've all seen pictures of the zombie ants induced by a fungal infection to die in positions that favor spore dispersal. Among parasitoids, most idiobionts simply use venom to paralyze the host and stuff it away in a burrow where the larva can feed in safety. But coinobiont parasitoids often manipulate host behavior in more complex ways, changing their responses to light, gravity, and other physical stimuli. The most common objective of these manipulations is to arrive at a safe location in which to pass the vulnerable pupal stage before the host is killed. For example, the phorid flies that decapitate fire ants first ensure they wander some distance from the nest so that the adult fly can emerge in safety. Aphidian parasitoids illustrate a variety of alternative strategies to avoid falling victim to intergill predation, which is always a threat within aphid colonies. Some species in the genus Aphidius induce positive phototaxis in the aphid prior to killing it. This generally leads the aphid out of the colony, which generally occur underneath leaves, and up to a high point on the plant, like a flower bud. While pupating in such an exposed location might seem like a bad idea, exposure to the sun will raise temperature and accelerate parasitoid development, shortening the length of the vulnerable pupal stage. Another genus in the tribe is Ephedrus. These take the opposite approach and induce negative phototaxis. Wasps of this genus form black mummies, which are well camouflaged when they form in crevices or cracks in tree bark, where they usually end up. The role of light in this process is easily demonstrated. When you rear these wasps on potted plants in a cage, the mummies all form off the plant, around the lower edges of the pot, and on the floor of the cage. But if the mummies form in darkness, they remain on the plants. The genus Trioxys is arboreal, parasitizing mostly aphids that live in trees. This genus takes an opposite approach to the Ophidia species. Their mummies simply do not adhere to the substrate, and they take their chances falling out of trees to the ground. Finally, Prayon has found a way to pupate safely within the aphid colony using crypsis. They leave the body of the aphid and spin a tent-like cocoon beneath it, leaving an empty husk of aphid cuticle as a decoy to deceive predators and hyperparasitoids. Idiobionts typically induce a permanent paralysis in their host, which not only protects them from injury, but can also be considered as a form of parental care. Coinobionts, on the other hand, need the host to continue feeding and growing, so their venom can take on a wider range of functions, but paralysis, if it occurs at all, is only temporary. Jewel wasp is a specialized parasitoid of cockroaches. There's a link to a video of the jewel wasp in action in the online resources section of this module, and a second video showing cockroach defensive behavior. The parasitism process has 10 discrete steps. It is a genetically programmed sequence of behaviors, as no learning is required. The roach is stung first in the thorax to disable its legs, and subsequently in the neck, which leads to longer-term paralysis, referred to as the hypokinetic phase. After clipping one antenna, the wasp tastes the hemolymph of the roach. It is thought she is sampling how much venom has been delivered and whether more might be required. The remaining behavior is quite typical of most idiobionts, except that the roach is not fully paralyzed until after it is left in the burrow. Long-term hypokinesis is also regulated via modulation of the thoracic ganglion neurochemistry. Wasps provided with a cockroach whose brain has been removed engage in prolonged periods of ovipositor probing, as sensory feedback is missing. All parasitoids have cincillae on their ovipositor that help them evaluate the suitability of the host during stings or ovipositor probes. So the act of stinging is always exploratory to some degree, and we should never assume that all stings result in eggs being laid. Because coinobionts must coordinate their development with that of their host, they can either take their developmental cues from the host or subvert the normal development. These alternative strategies have resulted in parasitoids being categorized as either physiological conformers or physiological regulators. Insect development is controlled by the action of two key hormones: the juvenile hormone, responsible for maintenance of juvenile characters, and ecdysone, which cues molting. And it is by responding to or altering the titers of these hormones that the parasitoid can either conform to or modify the development of its host. Obviously, there are a lot of potential advantages for species able to manipulate host development. Some parasitoids are able to induce as many as 6 additional larval instars in their host by preventing metamorphosis, thus obtaining more resources from additional larval feeding. This is common in gregarious species such as Cotacea. Extended larval feeding could be accomplished by interfering with production of juvenile hormone esterase, the enzyme responsible for degrading juvenile hormone. Certain ichneumonids carry a poly-DNA virus that causes degeneration of the host prothoracic gland, which is the source of ecdysone, thus preventing any further molting. However, other species may parasitize hosts that contain more resources than they need, in which case the host may be killed prematurely in an early instar, thus reducing the amount of time the larval parasitoid is at risk of predation within its host. A coinobiont must also balance its rate of consumption with the growth rate of its host so as not to kill it. If it hatches in a very small host, a common strategy is to remain in the first instar, which is a low energy demand, and wait for the host to grow before molting into the more voracious second instar. Consumption of host organs is not indiscriminate by any means, and organ systems essential for host feeding and digestion are typically the last to be consumed. Reproductive organs are usually the first, as their destruction has no impact on host survival. In the case of apterous aphids, the ovaries can constitute as much as 80% of total body mass. Aphidian parasitoids are short-lived, time-limited foragers that produce very small, yolkless eggs, which enables them to be produced in large numbers. But how does an aphid parasitoid, which hatches from a tiny, yolkless egg, overcome host defenses and manage to consume the entire body contents of its host? Aphidius urvi is one of the best studied species of aphid parasitoid. Here you see the earliest stages of the embryo's development, beginning with the first few cell divisions. Following rupture of the chorion in D, a serosum membrane forms around the early embryo, which serves two functions. It serves to shield the vulnerable embryo from the host's immune response, and at the same time channel nutrients to it so that it can grow. The serosum membrane surrounds the early embryo for about the first 120 hours of development. It has been removed in C and D on the left to reveal the elongation and segmentation stages of embryo development. The first instar larvae finally rips through the serosa in F, which is analogous to a second hatching. However, this is not the end of the serosa, as it dissociates into a series of large cells called teratocytes, which increase in size by a factor of 5. Their job is to break down the host embryos and deliver the nutrients to the developing larvae in a form of extraoral digestion. The larvae ingest what amounts to a nutrient-rich broth. The mandibles present in the first instar, which are used solely to eliminate competitors, are absent in second and third instars. Only after the entire contents of the aphid have been consumed, save for the nervous system, does the third instar guide its host to find a suitable pupation site. So what about aphid defense against this process? Because aphids survive on a very poor quality food, sugary plant sap, they have evolved with endosymbiotic bacteria that supply them with essential vitamins, sterols, and amino acids that they are otherwise unable to acquire or make for themselves. The primary endosymbiont of most aphids is Buchnera aphidicola, which is thought to have been acquired as much as 200 million years ago. This is now an obligate symbiosis, as neither aphid nor bacteria are able to survive without each other. The aphid even has special groups of cells called bacteriocytes, which function to house these bacteria. These bacteriocytes may or may not also contain any of 5 genera of secondary endosymbionts, which are not as reliably inherited as the primary symbiont and are not essential for aphid survival. Although the function of many of these remains unknown, possession of specific endosymbionts can enable aphids to exploit novel host plants, tolerate a broader range of temperatures, and in the case of Hamiltonella defensa, prevent the hatching of aphidian parasitoid eggs, providing partial to complete immunity to parasitism by this kind of wasp. Interestingly, the behavior does not confer protection against the eggs of aphilinid parasitoids, which have much larger, more heavily chorionated eggs, although it likely mediates competitive interactions between these two important groups of parasitoids. Among the most fascinating examples of host manipulation is bodyguard behavior, in which the host is left alive after egress of the larvae and induced to protect the parasitoid during the pupal stage. Some of these cases are turning out to involve endosymbiotic bacteria and viruses that are carried by the parasitoid, but have neural activity only in the host. In the case of Glyptopantales, as many as 80 offspring may emerge from a single host caterpillar, but one or two stay behind and sacrifice themselves as puppet masters. They force the caterpillar to remain next to the pupae of their siblings and cause it to thrash violently in response to any disturbance. Pupae that are not so protected by a caterpillar have significantly reduced survival. The bodyguard behavior of coccinellid adults parasitized by Dinocampus is more complex. Dinocampus is able to parasitize a wide variety of coccinellid species, although it seems to prefer North American species with which it has a long period of co-evolution. Regardless of species, the adult coccinellid is not killed, but remains alive, hugging the pupae of its parasitoid. This is a good strategy because lady beetles have aposematic coloration, which advertises their bad taste, deterring many predators such as birds. Experimental manipulations have shown that removal of the beetle reduces the survival of the parasitoid. If you're rearing lady beetles, you want to make sure you don't get any of these in your colony because they are uniparental, and one female can parasitize your entire colony without any need for mating. But the bodyguard behavior, it turns out, is actually mediated by a virus. The process is described in detail in the cited reference, which is provided in this module. The authors discovered an RNA virus which they called D. coccinelli paralysis virus, or DCPV. This is stored in the oviduct of female parasitoids and is injected with the egg at oviposition. It first replicates in the developing parasitoid larvae and is later transmitted to the host during development. Bodyguard behavior is brought about by replication of the virus in host nervous tissues, which induces an immune response, neuropathy, and paralysis, all the symptoms of the bodyguard behavior. In cases where the immune response is strong enough, the virus can be eliminated from the beetle and it can regain normal behavior, at least for a short period. Up until now, we've only looked at sources of mortality and how they contribute to biological control. But predators and parasitoids can have negative effects on pests without actually killing them, referred to as non-consumptive effects, a field of study that is often referred to as the ecology of fear. Herbivores can have their fitness reduced by engaging in various types of predator avoidance, as this is time-consuming and time they cannot spend foraging or feeding. In some cases, a herbivore may even be driven from a rich habitat into a poorer one. It is also possible for the behavioral responses of the prey to the threat of predation to have a greater impact on prey population dynamics than actual mortality inflicted by the predator. Most work focused on predators, but non-consumptive effects of parasitoids also occur. For example, parasitoids foraging in aphid colonies can cause many of them to drop to the ground, just as predators do. In diverse communities, non-consumptive effects can be additive among different predator species, including those that pose no threat to the responding herbivore, and they can thus have cascading effects on lower trophic levels. Many different developmental, behavioral, and even morphological changes have been observed in response to the threat of predation. For example, water fleas raised in water where predatory copepods are present develop longer apical spines for improved defense. Because of the broad importance of chemical communication among arthropods, many non-consumptive effects among them are mediated by semiochemicals. Many insects gain information about the risk of predation by eavesdropping on the pheromonal communication of higher trophic levels, just as parasitoids are known to orient to the sex pheromones of their hosts. This means that non-consumptive effects can extend some distance from their source and also persist in the absence of the predator. A lot of work demonstrating non-consumptive effects has involved exposing the prey to the stimulus while preventing actual consumption. And some have argued that this arrangement is biased towards an overestimation of impacts, especially in laboratory studies where organisms are confined. However, the power of this effect is well illustrated by the phorid flies that decapitate fire ants. The actual number of ants parasitized in the colony is very small, but large numbers of ants in the colony respond with defensive behavior when the flies attack, which severely limits the ability of the colony to forage and grow larger. So here is one case where a biological control outcome is mediated not by mortality, but by a non-consumptive effect. That concludes lecture number 9. In the next lecture, we will examine various contexts in which augmentation of biological control agents has been a successful pest management tactic.